11:30. Good morning again. And my name is Hongyu Miao. I'm an assistant professor at the Department of Biostatistics and Computational Biology. I'm now the director of the mathematical modeling core of the Center for Biodefense Immune Modeling. And I believe that after the talk of Dr. Hu Wu and Martin Zhang, you are all right now fully motivated, right? You are ready to do some modeling, right? Okay. So let's do it. So, but instead of using, languages or diagram, we start to do modeling using mathematics, So that will be the purpose of the lecture in this morning. So first, actually, I would like to ask the question: who is doing modeling? Of course, mathematicians, right? Engineers, we do that every day. How about the immunologists? My personal opinion, you are also doing modeling every day because you are using mice to study human, and many times that's by leap of faith, because we just believe that what we learn from the mice actually can be applied to human, So that's modeling. So what is modeling? My understanding, modeling actually is just the approximation, It doesn't really mean modeling has to be, mathematical or what. So modeling itself, by definition, my understanding is just the approximation. So when we're doing modeling, what will be the benefits we can get? First one, modeling itself is another way to represent our knowledge. I will actually have examples for each of the bulletins later. So for now, let's just go over all the points first. So second one, you can use it as a data analysis tool. You know that right now, one lab actually can produce like 10 times of the data than 10 labs can produce in the past. So within this huge amount of data, you need to find a way, systematic way to describe them, organize them, and eventually you want to extract useful information from this data. So that's what model can help. Also, you can use the model to do prediction and generate hypothesis. Sometimes I found that's actually quite easy, if I do that using model. However, in experiments, it's not easy sometimes, For example, I cannot, arbitrarily change the number of epithelial cells in the lung, so that I can actually look at the effect of available target cells on the virus infection, right? So, but I can do that in the model, just change the number, And, but you may not believe the results. I have to see that in advance, right? But it doesn't matter. We have a bunch of ways to check our model and check our results, which that's something I'm going to talk about later, actually tomorrow, Today I only have 3 hours, 1 hour in the morning, 2 hours in the afternoon, and I don't really have that much time to talk about all the details, but for now it should be okay. So the fourth thing is we have full control of our model, because you created it, right? You know every detail about the model. It's not like a biological system. Even you design the experiments, there are many factors we don't know, what's the role, they're playing. For example, the cytokines, what's the roles of all cytokines in the innate immunity? We haven't fully understand yet. But for the model, you already have this advantage because you simplified the problem, you approximate the real problem. So last one is cost effective. By saying this, actually, so we can actually use computer simulation, to help to design experiments and generate hypotheses and find out the most promising, direction for new experiments. So instead of randomly trying the combinations, there are an infinite number of combinations you can try in experiments. But the models sometimes can give you some clue, So also we're gonna actually learn the general types of models and more importantly, when to use what type of models, So models are like your culture or assays, It doesn't mean one assay can measure everything, Different types of models, you have to choose different types of models to solve different types of problems, Unfortunately, in the summer school, we're gonna only learn mainly we're gonna only learn the ODE and agent-based model, But there are actually tons of different models, you can use in practice. Also, at the end of this lecture, we're gonna learn some, very simple models and get familiar with the terminology, so we can be prepared to do modeling. So, back to the example. Okay. Here's actually the name I put from a web page. Okay, that's big companies. Okay, you know all the companies, right? They're actually doing modeling every day. For example, a car company. Okay, when they design a car, okay, they actually start to do modeling at the very beginning. Before they actually crash the car in the lab, they actually crash the car thousands of times. Okay, in the computer first. So that's how they come up with the optimal structure. Okay. That's why they can actually evaluate the safety, okay, beforehand, instead of design some defective, cars and later you change the design. That will be too costly, okay. So here's just some example. Second one, I want to talk about why we use modeling in biomedical research, okay. So I have actually an example for each of the 5 points. The first one is representation of knowledge, okay? So I think that we all have no problem with diagram, right? Diagram actually show us, this diagram actually shows us the infection or dynamics, okay? What's going on in the lung, okay, after primary influenza virus infection. This part we have, anyway, I cannot find pointer, but this is just so this one is actually our target cell. That's epithelial cells in the lung, and this is an infected cell. And we know that during the adaptive immune response stage, we have antibodies effect kick in, and also the effector CD8 T cells will play a role. By actually drawing this kind of arrow, okay, you can see a direction. This direction means from uninfected epithelial cells, it can become infected due to virus, okay, infection. So the diagram pretty much explains the story, right? By looking at the different components and the direction of the arrows. However, if we just do it in mathematics, okay, this actually is 3 equations. It is telling the exact same story, okay, here. We are actually just using a different language, okay? The biology is the same. So in that sense, okay, I would say sometimes you can actually use mathematical modeling as a tool to represent your knowledge, okay? That's fine. Once you know what's this language, okay, you will be comfortable with this language like you're comfortable with the diagram. So the second example is that actually Martin already talked about the CFSE data analysis, right? And instead of actually plot the marker intensity, okay, I just put up a number here, okay, because eventually, not eventually, you can actually skip some, intermediate steps. Anyway, this is data you can get from the CFSE labeling. At different time points, you can get a number of cells in different generation. Here, generation means the number of division. For example, there's the generation 1 means these cells never undergo any division, okay, during the time window of experiments. This one is after 1 division, okay. So you have this kind of experiment results. So can you actually, I'm going to ask the same question as Martin just asked. Can you actually tell the time to division or time to death just from this data? It's not easy because this data just tells you the number of cells at different time points, right? And how do we know, okay, the average time to division or time to death? So that's where we can actually use the mathematical models. This is actually the schematic illustration of the mathematical models we actually use to analyze the data. By using this data, actually, we can figure out the 2 parameters very interesting, okay, time to division and time to death. And under different stimulation, okay, that's probably what we're really interested in. Under different stimulation, okay, what will be the change in the time to division and time to death, right? Yes. Okay.
Are those numbers in generation 4, 5, and 6, those zeros, are those?
Yeah. you're supposed to.
After the 572, then you have 0, 0, and then you have more. So are those
Noise.
noise? Okay.
So
you just take them out
but
on your model.
if I have an error-free system, okay, I will expect that I see 0 here, see all 0 here, all 0 here, all 0 here, because there's a delay, okay. You need time to finish the first division and then finish the second division, right? However, due to error, you'll get this is a real number, okay. That's why you get error here. So now, something doesn't work. Jane, I got a warning sign. Battery is out. That's why. Okay. I'm gonna use the keyboard. Okay. Anyway. So the third example I want to talk about actually is how to use a model to do prediction and generate hypothesis. For example, okay, let's consider a new influenza virus string. Okay. What we actually want to answer is that if a neural string with high infectivity, okay, but moderate productivity, okay, will this string be more dangerous than a string with moderate infectivity but high productivity to infect the individual? Of course, I'm not talking about the epidemics, okay. So we actually did some, simulation based on the model we have. And surprisingly, okay, we had thought the infectivity should be the key factor. But later, when we look at these plots, if we change the infectivity, okay, actually for a few orders of magnitude, okay, the very low peak actually almost the same, remains almost the same. So which means if I increase the infectivity of virus, it only exhausts our target cell pool earlier, okay? And they cannot actually produce more viral particles, okay, within the primary infection. However, if I just increase the productivity, okay, for a few orders of magnitude, what you see is here, the peak of the viral load actually change a few orders of magnitude. So that's something, okay, some simple example. Once you have the model, have the parameter values, you can simply, manipulate the parameter values to see what will going on, okay, with the system. And this is a prediction, and actually you can match this prediction with the H1N1, okay, behavior, okay. That's exactly example, okay, of the prediction result. So now, after we know, okay, what the models can be used for, now let's look at what actually we can model, okay? So like I just mentioned before, there are tons of things we can model. At the molecular level, you can model the chemical reaction, for example, the interactions between the signaling molecule, okay, by doing molecular dynamics, okay. If you know something about molecular dynamics, you can use Amber, many packages, okay, just do a simulation. And you can also model the DNA mutation. For example, you can use this kind of thing to predict virus virulence. And also you can, of course, model the protein-protein interaction or drug-protein interaction. Okay. At the cellular level, of course, we are interested in the proliferation death, that kind of rates, right? Because under many circumstances, that rates actually can tell us lots of information. And activation, deactivation, and the differentiation class switch for B cells. At the organ level, okay, you can actually compare, okay, the contributions of different lymphatic organs. For example, the lymph node, the lungs, spleen, liver, bone marrow. In a recent study, okay, we actually compare the contribution of spleen with the contribution of lymph node, okay. So in that sense, you can identify which place actually is the major resource of immune cells, okay, to help you to answer that kind of question. Also, we can look at the interactions among organs. Basically, that's just migration and trafficking of cells, molecules, okay. Of course, if you are doing PDPD, okay, you get a blood sample. You want to know, because the data pattern will usually be up and down. Okay, from this up and down pattern, you want to infer the rates of the PKPD, and at the system level, I think that's a hot topic right now. Okay, at the system level, people are interested in signaling pathway, metabolic pathway networks, and you can do the gene regulatory networks. But of course, we know for this kind of large scale model, we also have lots of technique issue and also many inference problems. At the population level, we can do multi-species system. Like the simplest one is the predator-prey system, right? Just two variables, two species. And we can do epidemiology, social network, so that kind of thing. So sounds like I'm saying that we can model everything and we can do everything. However, I have to go back to experiments, okay? Modeling is just modeling, okay? Whatever results we get or predictions or hypotheses we generate have to be verified by experiments, okay? I'm not saying modelers can live by doing modeling alone, okay? Have to do it together with the immunologists, okay? So,
one question. So in this perspective, you were telling like prediction and hypothesis generation. Along with that, whether is it possible to do a model for experimental validation?
So you want to use model to evaluate
Yes.
your
like
experiments?
Instead of doing, say for example, high throughput studies, and further downstream we'll be doing some immunohistochemistry or something to do a selective list of candidate genes. So in such scenarios, instead of going for the immunohistochemistry or something, whether we can go ahead with the modeling to just to validate some of the candidate genes.
That's possible because we know that for the thing you just mentioned, okay, many times we measure that actually is too complicated. We hope that using this kind of measurement, we can understand the system, but we are not able to directly measure what we want to measure. Okay, so in that case, we can actually develop the mathematical models which incorporate different hypotheses and say which one actually can interpret the data best. And in that way, that's actually a way to compare different hypotheses and verify whether one of the hypotheses is the best one or the most accurate one that's supported by data. Okay. But that would be a very general answer, okay, I have to say. Because I don't have too much time to talk about every details or concrete example, okay, given the circumstance of the summer school, okay. It's more like, education. So, but anyway, you're welcome to, come talk to me, okay, after the lecture. So, here I want to actually give you, that's exactly the sort of thing you're interested in. Give you an example that what we're working on, okay. This is a big picture in our mind and we're still working on it, okay. So we want to actually understand the influenza infection, okay, within the past few years and also actually the vaccination and the adjuvant effect in the next few years. In our mind, we divide this system into 4 layers. Okay, we have genetic layer. That's the bottom one. We have this is a protein molecular layer. Okay, and this is the cellular layer, and the top one is the organ. Right now, we're not actually really moving on to the epidemics. Okay, so we focused actually on within host. Okay, immune response or vaccination. So at this level, you can definitely do some gene regulatory network modeling, and you can also construct a 2D, I call it 2 or 3D network. You can actually go through this, or cytokine, chemokine, whatever regulation factor. Okay, and then back to the gene network. Okay, and also we want to understand the cell kinetics. Okay, what do they do, and how fast do they do something, okay? So that kind of question. Open level is more like, migration, interaction, okay? Because we know many immunological phenomena actually they are localized, okay? Inflammation, okay, they're localized. They're not actually, everywhere. So that will be an example. Of course, that's just big picture, okay?
So is this a published one or unpublished one?
We published some papers for some specific problems. Okay, for the whole system, it will take lots of resources and time, okay, to finish. But I don't think actually we can finish this alone, because I think that's the job of the immunologists over the world, okay, not just the one center here, okay. So to model that many different things, okay, what type of model we can use, okay? Here we'll talk about different types of models. So in general, we actually can divide different types of model into two categories, okay? The first one is so-called deterministic models. The second one is so-called stochastic model. The so-called deterministic models, which means these models do not consider randomness. Okay, that's a key difference. But the stochastic model actually, they explicitly take the randomness into consideration. Okay, that's a key difference. So under the category of deterministic model, we have a so-called ordinary differential equation model, ODE. And we also have some variation of ODE, okay? For example, the delayed ODE. We also have something called a PDE, partial differential equation. So the difference between ODE and PDE is that PDE is more complicated. That's not a satisfying answer, right? The key difference actually is that ODE only can model well-mixed culture of particles, which means it cannot consider spatial difference. PDE actually can take the spatial difference into consideration. Okay, that's a key difference. But I'm not saying wrong things. Okay, PDE is much more complicated than ODE. We're gonna actually have some examples this afternoon. Okay, to look at these two things. So for the stochastic model, okay, there are actually too many different types of stochastic models. For example, branch process, okay, we know that it's actually, it was derived from, derived in like 1950 or 50s or 40s. But anyway, it's a long time ago, and people want to use this kind of thing to understand the cell cycle, okay. That's, actually, we're gonna have an example for this. Also, people use a Markov chain or hidden Markov chain, okay, not only to model the cell behavior or cell kinetics. You can also use the Markov chain model to model the sequence alignment, okay? If you're using, for example, BLAST or whatever, that kind of sequence alignment algorithm, okay? There's type of tools that actually use the hidden Markov model to do this. And there's something called the renewal process, which means a process can start and end and restart again. So that's called renewal process, okay? Queuing process is more like you have 10 customers, go to a bank, and but this bank only have 3, people can serve you, and then you have to wait in a line. And if you're interested in what's the average time to get served, that would be the queuing model. Okay, so this one stochastic differential equation, which means we introduce randomness into ODE or into PDE. Okay, so good thing is that now we extend ODE PDE to model, randomness. Okay, the bad thing is that whatever problems ODE PDE have, okay, the SPDE also have. Okay, and they have actually more problems than ODE PDEs if we're talking about how to use this model to interpret data. Okay, so but anyway, it's just probably take it as a personal opinion. Okay, so the last one is. You can also actually classify models based on your purpose, okay? For example, if I want to do molecular modeling, I can just use the molecular dynamics. I can also use the quantum mechanics, right? If I really want the accuracy, okay? Also, I can use different models to do network modeling. For example, you can use a Boolean network, Bayesian network, Rancher model, ODE. Also, of course, you can use ODE to model network. So, depends on purpose. You can also use, different types of models. So that would be too much to actually go through. Now let's talk about when to use what model, and the answer is actually disappointing because I probably give you too much hope, okay, at the beginning of this talk, but eventually the answer actually it depends. Depends on what? Complexity. The first one is. you don't want to use a very complicated model to model a simple problem. And also you don't want to use a very simple model to model a complicated problem, right? You have to match the complexity, okay, between the model and the real problem. So the complexity can come from the procedure, can also come from data. So procedure, for example, if we want to understand the T cell activation, okay, you know there's tons of steps, okay, involved, many factors involved. If you want to create a model for that kind of thing, okay, you do need a complex model. However, sometimes the problem may not that difficult, but data is. For example, if you want to understand study the HIV dynamics in patients, okay, the model we usually use actually only have 3 variables, okay, so three variables, 3 equations. However, we're gonna collect data from hundreds of, sometimes it's thousands of patients. So how to infer, okay, the kinetics parameter from a population of subjects? This time you have a complete data set. So in that sense, you also have to consider, okay, data complexity. Actually on Tuesday, that's where I'm going to talk about one example about this data complexity, okay. That's called actually mixed effect model, okay. And also you need to consider the characteristics of the problem. For example, if it's a dynamic problem, then you have to use a dynamic model. If it's a static model, a static problem, then you just use a simple static model. So I think there's actually a good example. Okay, when people do the gene regulatory network modeling, okay, you know the gene expression is actually a dynamic pattern. Okay, it's not static. It's not doesn't mean that we collect data on day 0 and day 5 and compare them, then everything is clarified. No, it's not that way. Because if you look at the data, okay, you have an up and down pattern. So you have to take that time course pattern into consideration when you model that. Anyway, okay, forgot to mention this. So sometimes you also have to choose whether you want to use the deterministic model or stochastic model. I leave that to tomorrow. Okay, this part. I'm gonna show you some example. Okay, when deterministic model actually is okay, and when you have to switch to stochastic model, and eventually, okay, even we do modeling every day, we have to try to modify. Okay, whatever you see in a publication, it's probably after a series of modification and refinement. So. Okay, so this is some cliche, okay? I have to mention every time. The first one is we talked about, okay, model is approximation to the truth. Second one, you can hear this everywhere, okay? Models are wrong, but some are useful. And I particularly figured out where it come from, okay? So models are wrong because they're approximation, which doesn't mean they are really wrong. They're wrong because they cannot take all the factors into the model. So if you want to write down every detail, okay, in your model, even for a very simple problem, for example, the infection in the lung, you will get hundreds of equations, okay? Use that model, you cannot do anything actually. So we actually sacrifice, okay, some unimportant details. And we just focused on those factors that we believe they're significant or important. So that's my understanding, okay, why people say models are wrong. However, since we captured the main factors, players in this game, we definitely can use it, okay, to interpret something. But to me, okay, I prefer to use the principle of parsimony because like I said, you can always create a very complex model, okay, as you want. However, to interpret your data, okay, I would prefer use the minimum number of equations or variables or parameters, okay, to interpret the data. So that also related to the identifiability issue, okay, Martin just mentioned. Tomorrow we're gonna actually talk about the identifiability issue. So for now, let's just say, okay, if we can use the minimum number of variables, parameters to well interpret a problem, then go ahead doing that. Okay, So I don't bother to, intentionally create a, very complicated model, and to model data. Sometimes I can give you trouble. Okay. So now, what's that? Okay. This one. I still have like 20 minutes. So let's do some simple math actually. Okay. If you're really interested in the mathematical modeling, okay, and you're not constrain yourself to what we teach here, okay. Here are 3 books you probably can read, and these 2 actually are from the same person, Marie, okay. And it's good. These are good books, but there are some typos, okay. If you can, tolerate that, okay. So next one, let's look at the simplest model, okay, for cell group. Now let's start to talk about numbers, okay. And when we talk about numbers, I need the chalk. Otherwise, I'm gonna be nervous. So let NT, okay, be the number of cells in the cell population, okay, at time t. So N here is a number, and this one is it's a function of time. Okay, that's the notation. So given a small time interval from t to t plus delta t. So delta t here is our time interval. Okay. We want to look at the change in the number of cells. So what mechanism we can actually consider? Of course, I can add the number of newborn cells, right? And also I can subtract the data cells. So they either were born during this delta t or dead, or become dead during this time interval. Okay. So although this one is very simple, but we're gonna use this, okay, again and again, especially tomorrow. Okay. So the basic idea is that we look at the change in numbers during a time interval. Okay, that's the basic idea. So that's how you actually derive the ODEs. Okay. This equation is sometimes also called a conservation equation. It doesn't matter, okay, what does it mean, okay, by conservation equation, I don't care. Actually, just sometimes if you meet this terminology in a publication or book, okay, you know what it is. Okay, that's all. And the specific form of the right-hand side equation, okay, it actually depends on what assumptions or what facts you want to incorporate into the model. For example, here I only consider the birth and death. If you also want to consider migration, you will have another term. If you also want to consider differentiation, you will have another term. Okay, so that's how you start from a simple model and get the one you want. Okay, by add more and more factors you want, or you believe that is significant. Okay.
Excuse me. So this conservation equation can be applicable to all biological processes, or not any restriction?
I don't want to say that because it will be too risky.
For instance, you said it's cellular proliferation and differentiation, but is it?
If we use ODE, okay, this sort of principle will apply. Okay, however, ODE also will fail. Okay, sometimes, and that's also something I'm going to talk tomorrow. Okay. So sometimes you cannot use ODE simply for some reason. Okay. So now we already have our first model. Okay, that's our first model in text. Okay, but let's now turn that text into mathematical expression. Okay, so how we do that? Now make it simpler. Okay, let's assume that there's no cell death within the time interval delta t. So we only consider birth. Okay. Now we use rho be the growth rate. Okay. Which actually is the number of division per unit time. Okay. That's the meaning of rho. Okay. The number of division per unit time actually per cell. Okay. That's one single cell. The number of one single cell can divide, okay, during the delta t. So, or unit time, not delta t. So now let's convert this first to mathematical expression. First, what's the number of divisions for birth during the delta t? Okay. that would be rho multiplied delta t. Because rho is number of deviation or bursts per unit time. You multiply the time length, then you get the number of deviation during this time period. Okay, is that clear? Okay, so however, at time t, we have how many cells we have? We have n. Okay, we not just have one cell. Okay, we have n. All the cells are capable of proliferate or divide. So that's why I also multiply, okay, n here. Any questions so far? Simple but important. Okay. So now I just convert the first term to mathematical equation. Okay. What's the next step? Next step, we cannot stop here because we know how to predict the number of cells in the population from the number at time t. But we cannot use this equation because it's difficult to use. If you want to calculate the number after 100 days, then I have to start from n0, and then I calculate n1, then I calculate n2, and here delta is 1 day. Right? That'd be difficult to use. So what people do actually, mathematicians do, is that we let this time goes to 0. So that sounds unreasonable, right? Because if the time goes to 0, then nothing can happen because the interval is too short. However, if we look at this, manipulation, okay, let's call it algebraic manipulation, okay, it actually makes sense. So this is the equation we got, right? In the previous slide, the first one. Now I remove nt to the left-hand side. So from positive becomes negative, you have to subtract, okay, nt. And then I divide delta t at both sides. So what you get is the change during delta t is equal to rho multiply n. So now delta t is gone, okay, from the right-hand side. Delta t only shows up in the left-hand side. Okay. So now if we let delta t go to 0, what's that? It's something we call the derivative. Okay. What is derivative? If we go back to this one, derivative actually is the change per unit time. Okay, that's derivative. The change per unit time, that's all. The rate. Okay. So just for convenience, okay, people use derivative to denote this kind of thing. Okay. As d and t. Here d means the differentiation, okay, operator. Or just you can just remember this, okay? This just means the rate, okay, the change per unit time. So in this way, we have, or we want to write it, I can write it in a different way. So you put a dot here, which also means derivative. Okay, that's the same thing as this one. And sometimes people also write this one as you can take this part out, okay? And this one means that you take derivative respect to time, okay? So you can actually separate them, okay? So d here means derivative or differentiation, and n here, which means where we want to calculate derivative. And in the denominator, again, you have this operator. D means it's part of the differentiation. And the t here is the variable you want to take derivative with respect to. Okay, so n respect to t. Okay, that's what it means. And for convenience, people also use this notation, n dot or n prime. It's the same thing. Okay. So they're the same thing. These three things, okay. So now, okay, I will be proud of saying we have our first model, okay, series model, and dndt is equal to rho multiply n. Okay, that's what we derived from here. Okay. So this model is also called the exponential growth model. Okay, before I talk about the exponential growth model, I want to further address what's the derivative. Okay, so if I draw a plot, so we know this number of cells is a function of time. It may look like this, okay, up and down. Okay, so given a time point, for example, t0, okay, what's the derivative here? The derivative here I can use just the slope of the tangent line. Okay. Remember, if I have a delta t, okay, that's our time interval, t0 plus delta t. So the length here is delta t. And the change in the numbers is here. So you divide this one by this one, you actually get the slope of the tangent line. Okay, so derivative, if you want to understand derivative in using graphics, okay, this will be the graphic, okay, interpretation of derivative. So the slope of the tangent line, okay, that's actually the rate. Okay, again, that's a rate, right? The change per unit time, that's a rate, same thing. Okay, now let's talk about this so-called exponential growth model. It's called exponential growth model because this model has a closed form solution. Okay. So not all the models have a closed form solution. Okay. This is just a very simple model. That's why it has a closed form solution. The solution of this model actually is okay, so you see a exponential here. So that's why it's called exponential model. Okay. And if you draw the plot, the solution will always look like this way. Okay, monotonically increasing. Okay, the solution doesn't matter what is your initial number of cells in this population, or it doesn't matter once rho is greater than 0. Okay, doesn't matter what you roll. Okay, it will look like this. Exponentially increasing. Okay, that's why it's called the exponential model. Okay, so now since we have developed our very first model, let's get familiar with terminology. Okay, so usually, okay, we have something called independent variable in your model. Independent variable means this variable does not depend on anything else. So itself, it will explain everything, okay? For example, the time t. Okay, we decided to measure the number of cells at day 0, day 1, day 2. We already fixed that number, okay? We know everything we are doing, okay? So t itself will not change once we design experiments, right? And that's why we call it independent variable, okay? It does not depend on something else. Yeah, however, the number of cells in the population we call the state variable, and sometimes we also call the dependent variable, because n is a function of t, which means n depends on t. And that is, n will change versus time. Okay, so that's why I call it the dependent variable. And the rho here in our model is called parameter. Okay, we talk about parameter all the day. Okay, that's a parameter. Okay, in the model. And initial condition, that's what I wrote down here. And also you can see it here. That's just the value of the state variable at time 0. Of course, this time 0 doesn't need to be 0, okay? It can be day 1, can be day 2, can be day 3, depends on where you want to start, okay? For example, I do not actually collect data at day 0. Instead, I would like to give it some incubation time. So I start to collect data at day 3. Then day 3 becomes my starting time point. And the value of the cells at day 3 is our initial condition. Okay, so I just put 0 here for simplicity. Okay, that doesn't mean the initial condition should always be 0. Okay, it can be any number actually. That depends on your question. Okay, so ODE model. Okay, if we give the initial conditions and parameter values, we can solve it. Okay, so if I say solve it, which means we can actually get this trajectory. Okay. If I have a model, I know the parameter value, I know the initial condition, we can solve it to get this trajectory. So that's the meaning of solving ODE equation. Okay, you will know every detail, okay, of this curve, every point. Okay, the value of each point on this line curve, you will exactly know the value. Okay, that's what it means. Okay, so we have talked about this closed form solution. Okay. That's same thing here, okay. Just there are some notation difference, okay. You write in this way, or you write in this way. They're the same, okay. They're the same thing, okay. So this is a power to a power of rho t, okay. It's the same thing. So, question so far? No, good.
So was rho? The parameters are something that you experimentally calculate, or there's a concept that you...
Here, parameter is more like a concept so far, okay? Which means we want to use some symbol to denote the growth rate, okay? We know there's something called growth rate in a model, and we denote that as a rho. However, the value can, of course, change. If we're looking at the T cells, and this value will be different from the value of B cells, right? And even for T cells, under different stimulation, okay, this rho will also change, right? For example, if you change the concentration of the cytokine, okay, the growth rate will change. So you have to plug in a specific value, one specific value, and to get one corresponding curve. And if you change this value, this curve will also change. Okay, does it make sense? Okay. So now here is some more terminology. I believe that you should already met this kind of terminology before, right? Doubling time, half-life. Okay. So when we publish modelers publish papers, okay, for many times we talk about half-life, doubling time. So what does it mean? Okay. So doubling time is the time interval for the size of the population to double. Okay. That's just the doubling time. Okay. So definition is simple. Half-life is the time of interval for the size of population to reduce to half. However, in mathematics, okay, the interpretation actually comes from these two sets of equations, okay? The first one is that we consider an exponential growth model, okay? Remember, we consider an exponential growth model. There's something called a linear growth model, okay? Later we're going to talk about. So it's going to be different from this one. So if we use an exponential growth model, we know the solution. We know the solution is this one, right? Closed form solution. Okay. So now we want to, after some time interval, now I let the T0 be the starting point, okay? And after a certain time point, T1, the size doubled, becomes 2 multiplied N0. N0 is our initial size of population. Okay, now it doubled. So after some calculation, you can just plug in, okay, the solution into the left-hand side. That's what you get here. Okay. Plug in the solution to the left-hand side. That's what you get here. And the 2 and 0 just come from here. Okay. Now you cancel out N0 because both sides have N0. And what's left actually is this one. And eventually you will find the time interval you need. That's the difference between T1 and T0. Actually is equal to log 2 divided by rho. Okay. So that's doubling time. That's how you calculate doubling time. Okay. Once you know the growth rate, So if you want to calculate doubling time, what you do is divide the row. Okay, log 2 divided by row. Okay, the two doubling time. So similarly, if you use exponential death model, okay, instead of growth model, so the difference here is that you have a negative sign. Okay, this is death. It's not growth. So the exponential death model will look like this one. So it will be monotonically decreasing, okay, instead of increasing, because we're talking about death. So the only difference actually is the sign change, okay, between the growth and death model. Now I do the same thing to this two equation, and eventually we found that surprisingly, okay, not surprising actually, because equations are too similar. Timing interval you need to reduce the size of the population to half actually is log 2 divided by t. Okay, that's what's called the half-life. Okay. Question? No? Good. So I probably are talking to very smart, class of students, right? So that actually makes me happy. Okay. So now let's talk about the properties of the ODE models. So once, again, I want to say that once the initial conditions and the parameter values are given, the solution is predictable at any time point behind t0. That's our starting time point with absolute certainty. Okay, that's what I just said here. Okay, you plug in one specific parameter value and give it a specific initial condition, then you will have a unique curve. Okay, remember that curve is unique. Okay, and you can tell every details about one data point on this curve. Okay, for example, what's its value? Okay, that's what we care about. So that's why it's called a deterministic model. Okay, again, that's our actually classification models. Okay, that's why it's called deterministic model. So also we have to note that the NT here is something called a continuous variable. So although we're modeling the number of cells in a population, okay, NT itself actually here is a continuous variable. So what could be the possible value of cell numbers in a population? 0, 1, 2. Can you have 1.5 cells? No, right? It doesn't make sense. However, when we do the modeling, okay, use the ODE model, okay, NT is a continuous variable. So NT actually can take the value like 1.5. And remember, the reason actually behind this is that NT is a mean. Okay, what we modeled actually use ODE is the mean number. Okay, is not the exact number. Okay, you can actually use stochastic model to model the exact number, integers, but here the NT actually is a mean. Okay, we know the mean of integers can be a real number, right? 1 plus 2 divided by 2. The mean of 1 and 2, it's a real number. It's not an integer. So that's why once you use a mean, you actually use a continuous variable. Okay. So this approximation actually works well. Okay, if your size of your population is large. Okay, even NT is a continuous variable. Okay, it will work well. Okay, if your population is large. However, if I say I have only like 5, 10 cells in my culture or whatever, that's time you need to consider stochastic model. Okay, then you will produce curves like this kind of discrete jump. Okay, instead of a continuous curve. So if you use NT, what you get is a continuous smooth curve. And if you use stochastic model, you can model integers, but the results will be like stepwise. So last question I asked here is, does it always work well? Since I asked it this way, which means the answer is no, right? So actually, yeah, tomorrow let's talk about some situations. Okay, the ODE will not work well, but fortunately, okay, for most of the cases, okay, in the study, you can actually use ODE. Okay, but there's just some special cases. Okay, you have to be careful about. So here I want to talk about how we actually make the ODE model work better. The first one is in our previous model, we say in a cell will exponentially increase without any constraint. That's not true, right? You have limited environmental supply, nutritions. So at a certain point, the growth should stop, right, and reach saturation, right? That makes more sense, right? So here, the first one I actually put up here is that ODE model structure is subject to change. If in your experiments or problem, you need to consider constraints or whatever. If my time window is very short, what I care about is a very short time window, I'm okay. I can use the exponential growth model because I know the saturation will never occur. I can actually use the exponential. But once I know that in my experiments I will get saturation, then I have to take this into consideration. So that's actually the model does. How this is the way this model actually considers the saturation by introducing one additional term here. So from here you can tell actually if n becomes n max, that's the max number you can get, the ratio becomes 1. 1 minus 1 becomes 0. So right hand side becomes 0, which means the cell will stop growing. That is the saturation, right? So the rate becomes 0, means cells stop growing, and you will expect to see a flat line, right? So the second one actually is a little bit tricky, so once you use the ODE model, actually you also made another assumption, not an assumption in your experiments or data or whatever. Actually, that's assumption in mathematics, and it's not actually easy to tell this implicit assumption. So the underneath distribution of the time interval for events to occur actually is assumed to be exponential in ODE models, like what we just talked about, It's assumed it's an exponential distribution for the time to event. However, once the time to event, does not follow an exponential distribution, you get into trouble. That's when the ODE models actually will fail. I actually have example tomorrow, talk about this. Finally, I think I'm over time. So finally, let's spend just like 1 minute, to go through the stochastic model, because that's really not my focus, Because tomorrow we're going to spend a whole day to talk about ODE, And Catherine will be the one who cares about the stochastics, So, but the basic idea is that now if I use the stochastic model, like I just mentioned before, I can actually directly model the number of cells, which means now I will model the integer, I will get an integer for the number of cells. And basically, the duration of this kind of model is based on the probability. So for example, at time t, what's the probability that the number of population is 1, is 2, is 3, is 4? So once you have this probability, then you actually specify the whole population or whole model. So again, if you're interested, you can take some time to read the slides, and the basic idea is the same, that we actually look at the changes in the cell number during a small time interval delta t. We still start from there. But how we deal with it actually is a little bit different. For example, if I have n cells at time t, I can actually have only one division. So if I have n number of cells at time t plus delta t, I can start from n minus 1 cells at time t, right? And then if I have only one birth, then I start from here and get here. So then we calculate what's the probability of this event. Similar thing. If I already have, sort of messy, but can you read? If it's already n cells are empty, then after a time interval, the cell number can remain the same. Okay, so I also take this kind of probability. Okay, so what's the probability that the cell number will remain the same? So after I calculate the possible probability, and then I can form this kind of sort of equation. Okay, so I calculate different, ways to get this same number, and then add the probability together, and you get the total probability. That's all. So that's actually the idea behind. Although mathematically it's a little bit more tricky. Okay, but the idea actually is simple. Okay, so I think I'm gonna stop here and see you this afternoon.